Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is going to be a very special EV club today for a few reasons. And the chief of those is that this EV club is featuring the special interest group of ICEV on extracellular vesicles in nervous systems. And so this special interest group is one of two that currently exist at ICEV. The other is the genitourinary system EV special interest group, or SIG. And these special interest groups are very important because they give an opportunity for broad sections of the community to come together and to talk about a specific topic that is of great interest to them. So this is our first EV club with EViNS. And it is also an interesting week here in Baltimore where I am, because today we're having a symposium of our extracellular vesicle core facility, and tomorrow will be the start of the ICEV X Tech meeting in Baltimore. This is the second iteration of this meeting that gives a platform for EV-related technologies and methods. And I hope to see some of you here in Baltimore later this week. I also am happy to announce that we've reached 5,000 signups for the EV Club, which started about 4 years ago, but it's grown quite a bit. The mailing list has grown quite a bit, and I can see that we have some new people on the call here too. If you have not signed up, if you're coming in for the EViNS event only, you're also welcome to sign up for a general mailing list. We'd be happy to have you here more often. All right, well, I would like to turn this over now to Tsuneya Ikezu, who is helping to lead EViNS, this new special interest group. And he's going to be hosting and moderating today. So Tsuneya, thanks so much for being here. I know that there are several others from EViNS, and I'm looking forward to hearing more about the work that's going to be discussed today. So thanks again.
Thank you, Ken, for this exciting opportunity to host the EV Club for the EVs in nervous system. So this EViNS special interest group has started by Dr. Christian Neri, who is also in a participant about a year ago. And then we are going to have the first journal club. And if you are interested in joining this SIG activities, I just pasted the website for this EViNS nervous system in the chat box. I hope you can access to that one, and so that you can take a look as well. So today I'd like to introduce the first speaker of the EViNS Journal Club series, Dr. Marcela Bartolio from Dr. Yonji Yan's laboratory at Tufts University School of Medicine. And she's going to talk about her very recent work, which is recently submitted to the bioRxiv for the preprint. And the title of the talk is glutamatergic regulation of microRNA containing exosome precursor trafficking and secretion from cortical neurons. So Marcela, please take a stage.
Okay, thank you so much, Tsuneya. Ken, I'm going to share my screen now. Thank you so much for this opportunity to present this work. First, well, my name is Marcela. I'm from Argentina. I did my PhD in Argentina, and then I joined the Young's Lab 3 years ago. This was my first project in the lab. And just to talk briefly about the Young's Lab, we are a neuroscience research lab that is located in the Tufts Medical School campus in Boston. And we have some projects related to exosomes. Some other projects are not involved with extracellular vesicles, but I would like briefly to mention one of the, I would say, biggest projects in the lab, and that was recently published as well. In this case, is related to the astrocyte to neuron communication, and this paper was published last year in Nature Communications. And in this case, it was described how the exosomes from the astrocytes and the hepacam protein in these astrocytes can stimulate the axon growth of the cortical spinal tract axons. And this is possible because of the ApoE is decreased in the first week, postnatal week. But then in week 2 or 3, the ApoE increases its expression and can antagonize this effect on the axonal growth. but in turn, it can stimulate the dendritic spine growth. And then related, and this is happening on the development of the layer 5 cortical pyramidal neurons. And related to this paper, there is a new one that is going to be published next week, was accepted already. And this one, it's about how pro-inflammatory cytokines induce the loss of neuroprotective function of astrocytic exosomes in an SOD amyloid lateral sclerosis model. So you can see also in this work, you will see how these exosomes coming from reactive astrocytes can antagonize the neuroprotective effect of the HEPA-CAM protein from these astrocytic exosomes. So then I like to mention some other ongoing projects related to the exosomes. One is the role of CD63 in regulating the microglia tau clearance through intracellular degradation and exosome-mediated secretion, and also the secretion and parenchymal dynamics of exosome from reactive astrocytes of Alzheimer's disease mouse models. These projects are conducted by Sean Hacker and Carly Reynolds, respectively. They are both PhD students in the lab. This is, as I said, this is the first work that I did in the lab that involves the glutamatergic signaling in neurons and how they can regulate the intraluminal vesicles and mirror content and the secretion of the cortical neurons. This paper is available now as a preprint in bioRxiv. So as an introduction, we know that extracellular vesicles are communicators between cells. And in the CNS, all cell types, the main cell types, can secrete extracellular vesicles. We can find mainly two types of these vesicles, which are the membrane shed microvesicles and also the endosome-derived exosomes that comes from the multivesicular bodies in the cell. Undoubtedly, the exosomes are the most studied type of EVs right now, and specifically in neurons, we know that they are involved in some processes, like, for example, in development and proliferation, in the neurotransmission, and also neurodegenerative diseases. In this case, they can have protective and also toxic roles. And I like to mention also the neuron-derived virus-like particles. These are type of vesicles that are formed by the ARC or the PHE10 proteins. These proteins are gag-like proteins that can self-assemble into virus-like capsids, and they can be secreted in vesicles from the neurons and be transferred to other neurons, transporting messenger RNAs. So we know that the origin of the exosome is in the MBBs of the cells, and in the neurons it was described that in soma and dendrites, we found like almost 50 times more MBBs than in the axons. And in these EM images, you can see these two corresponds to soma of the neurons. You can see the MBBs here with the intraluminal vesicles. Here are dendrites also showing MBBs. And this is an axons with the myelin sheath here. And also we can find an MBB in the axon. So this can also, we could also assume that the secretion of the exosomes is taking place mostly in the soma and in the dendrite, although the trafficking is not very well understood yet. Now, as part of the cargo of the exosomes, we have the microRNAs. The microRNAs are a type of non-coding small RNA that the function is to inhibit messenger targets in this case, on the recipient cell, because they are traveling through the exosomes. Here you have some examples on how some microRNAs delivered from neurons can affect the recipient cells, like, for example, microglia, where the miR-124 can attenuate neuroinflammation and also play a protective role after the brain injury. In astrocytes, the miR-124-3P regulates glutamate transporter 1. Also, there is function of miR-132 regulating the vascular integrity. And from neuron to neurons, it was demonstrated that the brain-derived neurotrophic factor can induce the specific sorting of these three microRNAs into extracellular vesicles, and then this is transferred to hippocampal neurons where they can form excitatory synapse. So we can see also, yeah, and this is like also a common knowledge that this cargo, the microRNAs and also the proteins that are containing extracellular vesicles can change according to the, like the context or the cell activity and beyond. On the other hand, it was also demonstrated that the release of neuronal exosomes is closely related with synaptic activity. We all know that the glutamate is the principal excitatory neurotransmitter in the central nervous system, and a couple of works have demonstrated that the synaptic glutamatergic activity in neurons is inducing the release of the exosomes. In these investigations, they did in vitro cultures of neurons, and they were depolarized using potassium chloride or GABAergic antagonists, and they measured the secreted exosomes based on marker immunoblots that we know that maybe that's not the best way to accurately measure the quantity of secreted exosomes. And although all these papers, the contributions of these papers are valuable, we still have questions to be addressed or questions that are unanswered. And here is one is, does the neuronal activity regulate the in vivo neuronal EV secretion? As I said before, this was also shown, this was all shown in vitro, but we don't know what is happening in vivo. The second question is if the glutamatergic signaling can alter the microRNA packaging into the intraluminal vesicles in the neurons. And third question is the subcellular localization of the intraluminal vesicles affected by glutamate stimulation in neurons? So those are like kind of the questions that we try to answer in this research. So to do this, previously in the lab, we generated this intraluminal vesicle exosome reporter mouse model. This animal harbored a flock stop codon upstream of the human C63 GFP fusion protein. So when you cross this animal with some Cre expressing line, you can get this stop codon removed, so the human C63 GFP will be expressed in a cell type specific manner. I'd like to highlight that this animal is available in Jacques' lab now. So to start with this research, we have here cultured neurons in vitro. This virus was added, the AAV9 KMK2 Cre, to produce the human C63 GFP. And the first thing that we notice is that it has a perinuclear localization and also overlaps very well with endogenous mouse C63 that you can see here in red, and the colocalization in yellow here. And in these EM images of the cultured neurons, you can also distinguish the MBB and the immunogold labeling of the GFP. Some signal is detected in some of the vesicles inside the MBB. And then we did the treatment with the glutamate in culture. We treated for 4 hours at 37 degrees, and then we analyzed again the colocalization of the human C63 GFP with the mouse C63, which it's slightly changed, but not so much. And we also measure the markers by immunoblot, the human C63, the GFP, the mouse endogenous C63, and also CD81 that we know it's also an exosomal marker, and we didn't detect any change on the expression of these markers after the glutamate treatment. And this is the densitometric analysis showing that there's no change in the expression. But interestingly, when we measured the distance, the mean distance of the GFP positive vesicles between the center of the nucleus, we could see that after the glutamate treatment, that distance increases, meaning that there is like a scattering of these intraluminal vesicles, GFP positive particles in the soma. And this is also consistent, this result, with the finding that we have a reduction of human C63 positive, GFP positive punta in the soma, and an increase in the neurites. So because we detected previously that the proteins are not changed, the expression is not changed, this is not due to an increase of the expression of this marker, but it's an effect of the glutamate in the pre-existing vesicles in the soma. So we further, because C63 is a well-validated intraluminal vesicle marker and exosome marker, we wanted to explore the subcellular localization by doing immunostaining of the endosomal pathway in this case, and we use EA1 marker for the early endosome, RAF7 as a late endosome and MBB marker, and we also included a Golgi marker because we also know that the ER-Golgi network can deliver newly synthesized protein directly to the compartments, the endosomal compartments. And we could see by analyzing the colocalization of the human C63 GFP with these markers that it's predominantly overlap with the RAF7. So yes, we could see like the overlap is predominantly in the RAF7 marker, which it's indicating that this signal, these particles are associated probably with multivesicular bodies. And then with the EA1, there's not much colocalization. And we have a little bit more, like 15% colocalization with Golgi. That could mean, so sorry, after the glutamate treatment, what we notice is that the colocalization with RAF7 is reduced, but it's not affected the colocalization with the other markers. And we also measure the protein expression of EA1 and RAF7, and also observe that EA1 is reduced after the glutamate treatment and RAF7 is increased. So this is telling us that, well, the endosomal pathway is being affected by the glutamate. Probably this is due to the transition of this type of endosomes, you know, from early endosome to late endosomes. And also probably the increase in RAF7 may be implicated in maybe more lysosome function. But anyways, those are like just hypotheses here. So as I said before, because we know that maybe the Golgi can transfer proteins directly to the endosome, the endosomes, we also did immunostaining of the RAF7 with the Golgi marker, and we could see that the colocalization is also around 15%, which is matching with the colocalization between Golgi, the Golga5 and the human C63 GFP. So we can say that a portion of this newly synthesized human C63 GFP is trafficking directly from Golgi to the network of the MBBs. And there's no change with the glutamate treatment. Now, the next question was about the secretion of the extracellular or these exosomes. For this, we collected the neuron conditioned media from control cultures and also the cultures treated with glutamate. And after size exclusion chromatography, we isolated the exosomes that then were quantified, and the size was measured by the CeraView nanoparticle tracking analysis. The first thing that we can notice here is that the size distribution is very similar between the two groups, but we could confirm that after the glutamate, there is near 45% of increase in the secretion of vesicles in the glutamate treated, which confirms the previous findings, but in a more accurate manner. Then we also detected the markers of the exosomes. And you can see here the human C63, the GFP, the mouse C63, also CD81, ALIX, and we also tried the ARC protein that I mentioned at the beginning. We didn't find any signal of ARC here. And we also did the CD9 marker that cannot be detected on these EVs. So this immunoblot results is supporting the idea that these exosomes or vesicles, secreted vesicles, are coming from the multivesicular body and not from the plasma membrane because of the lack of CD9. So next, we examine the microRNA content of these exosomes after the glutamate treatment. And here is the distribution based on the mean count of the total microRNAs detected by the small RNAs experiment. And we didn't see, I mean, the pattern is highly similar between the control and the glutamate treatment, and also we noticed that there is highly expressed microRNAs that are highly conserved between the control and the glutamate treatment, and you can see those microRNAs here, that they are highly conserved. And then in terms of the differentially expressed microRNAs, we found 18 upregulated and 5 downregulated, but more than 700 microRNAs are unchanged by the glutamate treatment. So maybe we can say that the MIR they were not so altered, the MIR cargo in this situation. So we decided to continue to evaluate the abundance of the highly expressed microRNAs in the neurons and also in the neuron-derived extracellular vesicles. We also included the MIR124 here because although it was not on the top highly expressed microRNAs, we know based on previous study that these microRNAs can be transferred through exosomes from the neurons to the astrocytes. So with qPCR, we measure the quantity of these microRNAs, the MIR9, MIR125, MIR124, and MIR128 in the neurons of control and glutamate-treated cells, and also in the exosomes derived from these neurons, from the control and the glutamate stimulation. And as you can clearly see, the treatment is not affecting the expression of this microRNA in the cells, in neurons, or in the exosomes. But then when we calculated the relative enrichment of these microRNAs in the extracellular vesicles, we found that there is like a selective packaging of some MIRs. For example, MIR9 was highly expressed on the neurons, but only modestly packaged on the EVs. In comparison with, for example, MIR124, that you can see it's not so expressed in the neurons, but when we do the ratio or we calculated the enrichment in the exosomes, we could see that it's highly enriched in the exosomes. So this is supporting also the notion that some microRNAs are selectively packaged into intraluminal vesicles. And to further illustrate that this microRNA was being packaged in intraluminal vesicles, we transfected neurons, these human C63 GFP expressing neurons with the sci-fi label MIR124. We stimulated with glutamate and then analyzed the colocalization between this label MIR124 with the human C63 GFP signal, and we could see that almost 70% is colocalized. So like kind of supporting this idea that at least this MIR124 is being packaged in the intraluminal vesicles and then secreted. Now, to demonstrate what's happening in vivo with the spreading of the exosomes and the activation of the neurons, we use the DREAD chemogenetic approach. So this is an engineered receptor. In this case, it's called the HM3DQ. This is an excitatory type of receptor that it's only activated by a specific compound that is called CNO. So what we used was here we use the human C63 GFP model, and we injected on one side of the brain the synapsin Cre virus, because, well, one thing is that this receptor is Cre dependent, and also we know that our animal model is also Cre dependent. So we need to add the Cre recombinase. In this case, we injected Cre on one side of the brain. This is the control side. And on the other side, we did a combination of the Cre virus and also this HM3DQ chemogenetic receptor. So what you will get is that one side will express only the human C63 GFP, and the other side, you have the mCher expression that is fused to this engineered receptor plus the human C63 GFP. After adding the CNO compound, we measure the activation by immune staining with CFOS, and we quantify this CFOS staining. You can see here in this image that the CFOS is highly detected on the side with this receptor and not in the other side. And then here, by quantifying the CFOS staining, we can also see that the control side is much lower than the side expressing this receptor. This is the CNO administered group, and we also had a group without CNO. that we can also see that there's no change in the human C6, in the CFOS immunostaining. So after this, we then measure the GFP, the C63 GFP positive tissue area on the injection site section, and also a section more distant from this injection site. And we noticed that the area of GFP positive tissue is larger in the site that is expressing this engineered receptor, meaning that the activation of the neurons by this receptor is promoting this spreading of the of exosomes in the brain and at distal sections as well. And this can also be seen in this graph here, where you have the injection site and the comparison between the control side of the brain and the site that was injected with the engineered receptor, how the tissue, the GFP positive tissue area is larger in this site. This is not happening in the animals that were not injected with the CNO compound. So these results corroborate that in vivo, the neuronal activity also promotes this EV secretion and spreading in the brain. So then to further illustrate, because in vivo localization of intraluminal vesicles remains like unexplored, we decided to look for in situ localization of the human C63 positive intraluminal vesicle from some neuronal subtypes by generating triple positive mice that can express the TDT in a specific manner in the neurons, and also the human C63 GFP in that neurons. We've done this experiment before in cortical neurons, in excitatory neurons. So this time we choose the motor neurons in the spinal cords and also the dopaminergic neurons in the VTA. So for this, we have these the motor neurons are the HV9 neurons. And this animal is expressing the CRE specifically in these neurons. So by crossing these animals with AI14 and with our C63 GFP mice, we get the expression of TDT in the neurons, in the motor neurons in this case, and also the human C63 GFP in the same neurons. And this is the same thing for the dopaminergic neurons. In this case, the DAT-CRE animals was used, and we can see expression of TDT in the dopaminergic neurons and also the human C63 GFP being expressed on this type of neurons. And also images were taken at different time points. In the case of the motor neurons, we have images from embryonic day 12, embryonic day 18, and also P7. And then for the dopaminergic neurons, we started in E18, P20, P40, and then we evaluated the colocalization between the human C63 punta and the TDT of the astrocytes. And we get around 80% of colocalization, meaning that the remaining 18 or 20% is GFP that is alone. So we can say that that punta was secreted at some point during the development of the animal. So finally, to further illustrate more higher resolution what's happening in the synapse and close to the blood vessels, we generated this virus that is expressing DIO human C63 GFP with synapsin CRE promoter. This was injected in the VTA of the DAT-CRE mice to promote the expression of this human C63 GFP specifically in the VTA. And then we did EM analysis by immunolabeling the GFP and a quantification of these immunogold positive particles in axon terminals and also in dendritic spines. What we found in the D18, in the dopaminergic neurons, is that the signal of the immunogold was 63% positive in the axon terminals and only 25 positive in the dendritic spines. And this is the opposite that happened in our previous study with the excitatory neurons from the forebrain, where we found more signal on the dendritic spines. And in the EM images, You can visualize here, this is the axon terminals that is full of presynaptic vesicles. And here in red, this is the intraluminal vesicle with the immunolabeling here. And then in this case, it's also an axon terminals, and we can see here there is a blebbing of the membrane. And we can see right after this this structure, we can see on the other side, there is also a vesicle with the immunogold labeling here. So this could be the transference from one side to the other. And this is also depicting here the immunogold in dendrite spines. And here, this is an image where we can see a vesicle with immunogold that is near the blood vessel lumen. So, okay, that was fast, sorry. Here, so with all this work, we can have some main conclusions here. The first one is that we could corroborate that the stimulation of the EV secretion by the glutamate is increased, and we did that. Sorry, I entangled. So the stimulation of EV secretion by glutamate was quantitatively measured, providing a more accurate assessment of EV secretion compared to methods based on the protein markers. That was the work done with the NTA analysis. Then the neuronal exosomes are primarily MBV-derived rather than plasma membrane-derived microvesicles. We also show that the neuronal excitation increases in vivo spreading of the neuronal exosomes. The last work with these DAT neurons and the HV9 in early stages also support this cell-cell communication, and that could be really relevant during the development as the neural connectivity has not formed yet. So maybe there's more, they rely more on this exosome communication in early development. Then specific microRNAs are selectively enriched in neuronal EVs. And lastly, the subcellular localization of intraluminal vesicles vary among different neuronal subtypes. And this suggests some functional diversity in exosome trafficking and secretion. So in some future directions, well, maybe explore more the glutamate effect in another type of cargo, like the proteins in the exosomes of neurons after the glutamate stimulation, and also investigate the mechanism of microRNA sorting into intraluminal vesicles. And I would like to add that probably it's going to be really viable to do in vivo also analysis of the exosome, the secreted exosomes after activation. Is more complex, but I think it's also the next step. And lastly, additional studies are needed to try to figure out how the EVs are secreted in neuronal subtype, different neuron subtypes, and also the involvement in development and also pathological environment to understand more the CNS communication and disease progression mediated with the exosomes, by the exosomes. So, okay, yes, I would like to, of course, thanks my lab, the young lab, to Yonje as well, and all the lab members. They are awesome. And also I want to thank the Wu lab because they collaborated in this work too. So I think that's it. Thank you so much for listening. And I'm open to questions or discussion.
Thank you, Marcel, for the excellent presentation. So the floor is open for questions. I just you can unmute yourself and then start your video if you are interested in.
I have a question. This is truly exciting and important work for the field. And my name is Shinichi. I'm from University of Alabama, Birmingham. And then you mentioned that those like dopaminergic like neuron EVs are basically facing the bloodstream lumen. And did you detect EV like component like a human CD63 signals in the blood EVs when you collect blood after like injection of like this virus, or even like after CNO stimulation of neurons?
No, well, we didn't collect those samples. That experiment in vivo was also done in the tissue, and that was it. We didn't do the collection. Of course, that's something that we try in other projects to look for that CFP signal, the labeling in circulation. Yes, actually, the project that I mentioned at the beginning that related with Alzheimer's disease and the peripheral circulation, that's kind of also looking for the CFP signaling outside of the CNS. But no, in this one in particular, we did not isolate exosomes or vesicles from the blood.
And another question is, what is a kinetics? You did also cell culture, like neurons also like secrete synaptic vesicle, obviously, right? And then compared to synaptic vesicle, like how fast like EV secretion is when you stimulate with glutamate. Do you have any?
What's the question again?
How first is the secretion of EVs? Like
well, I tried different.
Yeah.
Well, we know that the secretion of the synaptic vesicles is fast after the simulation, but in case, at least in my experience with these cortical neurons in vitro, I tested different time points. I started with 5 minutes because I thought, okay, maybe it's gonna happen really fast. So I did 5 minutes and I couldn't detect any difference, like in terms of the secretion. I tried 15 minutes, an hour and a half, and then at the end I choose 4 hours. And this is also something that was used in previous papers with when they tried the polarization with the potassium chloride. They did, I think, 3 hours or 4 hours. But that's something, yes, I could see that difference in the number of extracellular vesicles or exosomes after 4 hours. Like that was really increased. So yeah, I can say that it wasn't as that fast. I thought it was going to happen maybe faster, but no.
Very interesting. Thank you.
Okay, thank you.
So Eva Maria has a question.
Okay. Hi, Marcela. Thank you
Hi.
for that great presentation. I really enjoyed that. Also, your in vivo models that you implement. I think that's very important. So I have actually a few questions, but I limit myself to 2 now for the first round at least.
Okay.
First question, very simple. Can you stimulate in your in vitro experiments, or not stimulate, can you interfere with exosome secretion or EV secretion by applying glutamate receptor antagonists? First question, maybe first answer, and then maybe I can have my second question.
Yeah, well, that actually was an experiment that I also wanted to do because it was like a complement for the glutamate stimulation. What if we block the NMDA and the AMPA receptors? Unfortunately, I don't have that experiment. It's something that can be done, and actually something that other people did before. They block the AMPA and the NMDA receptors, and that's why they concluded that glutamatergic synapse was involved in the secretion of EVs. So yes, that can be done. Yeah.
Okay. And then my second question was, when your experiment where you show the spreading of the EVs in vivo in the brain with the viruses that you inject, on the IPSI and contralateral side, on these 2 sides. So you say the signal is spreading, but how can you be sure that what you look at or what you measure is EVs? I mean, it could be CD63 GFP expression that somehow gets activated from the construct with the activity, or that the virus is spreading, or how can you make sure that what you see is EVs? Do you get what I'm?
Yes, that's a good question. So yes, because we compared both sides of the brain, we can see here the side with only the pre-virus expression. And yes, we can see that the C63 expression is happen, of course, in the injection site and also on distal areas. But in the case of this injection alone, we see like it's reduced when you go far away from the injection site. And that was not the case when we stimulated this receptor with the CNO. So for example, in this case, you can also see that the receptor here is present with the mCherry, but at this point, there's no more expression here of the receptor. So, but we still can see a lot of CD63 being expressed in that area. So in comparison to the other area. So we could say, yes, maybe some part of it is because the Cre virus traveled to that distant area, but the excess of expression, we believe, is because of the spreading that is being promoted by this specific activation. I don't know if that answers your question.
Yeah, partly. Yeah,
Okay.
but okay. Yeah. Yeah, thank you.
Okay.
Thank you. Yeah, Christian, please.
Great talk. Thank you so much for sharing this data and explaining us. Maybe I missed something, but did you look at what happened in the striatum? You know, whether there is a signaling between the cortical neurons and the striatal neurons? I am
where
unclear
exactly?
about, yeah, on this slide. Yeah. Could you comment more on what you see in the striatum, in the dorsal striatum, or?
You mean here in this area?
Yeah.
Yeah, I mean, we analyze the area of the GFP positive. This is the injection. I cannot say if this is an effect of the injection or if it's transferred directly from the cortex to the striatum. The injections were done in the cortex. So yeah, maybe we were not expecting to have a signal here in this area. That's true. But yeah, I cannot explain. I mean, it could be an effect of the injection of maybe the spreading of human C63 GFP in this case.
Okay, thank you.
Yeah.
So I have some question actually. So related to Christian's questions, if you use the DAT Cre mouse model, you specifically express this construct in dopaminergic neurons. Did you see the spread of the EV to the coded nucleus? It's an axonal projection from that region.
In this experiment,
DAT Cre mouse? Yeah, you express nicely in substantial nigra. And then usually this neuron projects the axons into the coded nucleus, right? So did you check that? I was wondering how the EV is spread. Is it spread locally from cellosome or spread through axonal terminal? So I wanted to know what is the behavior of the EV spreading from EV secreting neurons in your experiment?
Yeah, well, no, we didn't check that. We took the EM images from the VTA, which has focused on synapse here, but I cannot specifically address that question in this experiment based on what we did.
Maybe you can do some immunohistochemistry of the striatum using this mouse model, and if it's positive, you can do immuno-EM in that region for validation, for example.
Right. Yes. No, those are good experiments that could be done. Yes, of course. Yeah. To better, have more information about this secretion. Yeah.
Okay. Thank you.
Thanks.
Any other questions?
Hi, let me ask a quick question. So I'm a neuroscientist and I know a little bit about these EVs. And so very interesting data. I like your mechanistic type of work, and you did in vitro and vivo stuff. So maybe you don't know this, and just ask a question about your thoughts. So you show that with glutamatergic activation or activity, you don't see so much of a change in profile in microRNAs that are detected in EVs or ILVs. You see just the magnitude. But then what you found cell specifically was sort of the location of these ILDs in dopaminergic neuron versus glutamatergic neuron. Do you think that the difference in, portfolio of these microRNAs would be different, cell specific? Dopamine neurons would have a different portfolio of these microRNAs versus glutamate neurons would be different. So you see that will be sort of a difference you might expect.
Yes, that probably, yes, you can find differences. That's one of the characteristics of the extracellular vesicles, that they are also the cargo is also cell type specific. You can find probably in the case of the microRNAs, maybe you can find different profiles of microRNAs in different subtypes of neurons, and that's something that also, it remains to be investigated. But yes, I believe that yes, that's probably it's going to happen.
Are you going to look at that? Look at dopamine profile versus the glutamatergic versus dopaminergic neuron profile?
Well, it's not planned, but of course that would be like a nice line
I'll be
to
interested
follow.
to know. Yeah.
Yeah, because like in general, yes, we are like interested in evaluating more about the subtype of neurons in terms of the exosome secretions. That was like the main focus, but of course it's tightly related to the content of those exosomes. So if we could accurately, because I'm thinking like in vivo, if we could like separate the vesicles from different neuronal subtypes and then analyze the content, I think that would be like really, really valuable information. But in vitro probably can be done as well. Yes.
Yeah. Thank you.
Yeah. Thank you.
So actually, I have a little bit more question, but maybe I can ask us the next question only. Go ahead.
Hi, I have a short question. So you know that there is an increase in release of these vesicles after stimulation, right? So how about, is there is a way in your model to look what kind of cells are main recipients of these extra vesicles which released after the stimulation? What do you think about that?
That's also a good question. Like, okay, where are these exosomes going? So here, the only thing that I can show you, because I didn't add it just to not make the presentation longer, but we did here in this graph, Here, what we did, this was a prediction of where these exosomes may be going based on this microRNA content that we found on the top, the highly expressed microRNAs. So based on the targets of the predicted targets of this microRNA, we compare with transcriptomic data on the different cell types here, that you have the neurons, endothelial cells, microglia, astrocytes, or oligodendrocytes. And then by like comparing the targets for the microRNAs and the most enriched proteins in these cells, we potentially can say that, for example, MIR128-3P, this could be delivered to neurons. In this case, could be going to microglia because of the target, because the targets are more in this case, and in this case is endothelial cells for the MIR9. But this is based on microRNA. Of course, we need more studies to really figure out the destination of these EVs.
So do you think there are different type of EV secreted after the stimulation, which could be actually, like destinated, have a destiny to go to different type of cells?
Yeah, I think that was something that, there's a discussion about that. Like, for example, is an MBV producing one type of intraluminal vesicles, or it's a mixture of different type of intraluminal vesicles, and then, after secretion, they can go to different cells, recipient cells? I think that yes, based on what I've been reading, I know there's still a lot of work to do regarding that, but yes, probably they can secrete and send messages to different cells that can act in different manners. In this case, regarding the glutamatergic activity. Yes, I mean, probably they can message neurons and also astrocytes, to help in the cleaning of glutamate or things like that. Yeah.
So maybe like surface proteomics would help, right, for those EVs to
Yes.
see
Yes.
where they're going.
Yes,
Thank you
for
very
sure.
much.
Yeah. Yeah.
Yeah, excellent talk. Thank you.
So I try to understand this data. So you did primary culture of different cell types and you apply the neuronal conditional media before and after the glutamate stimulation. And you check the microRNA profiling in the recipient cells. Is that the data?
Here, no. This is all a prediction. Here, what we did based on the microRNAs that we found, we perform the list of the predicted targets of these microRNAs that, that information is found on TargetScan or MIRCHAR base, and then we compare those targets with a transcriptomic database of the different cell types and try to see, targets. We have the list of targets of the microRNA and the list of proteins that are enriched in the specific cells and see, overlap them and see potentially which MIR could be targeting messengers and in which cell could be the potential, recipient of this. Yes, but we didn't do this in vitro, but that could be like a nice experiment too.
Okay, thank you. Questions? Okay, if there's no more questions, really thank you for the excellent question and the presentation, Marcela, and we look forward to see your paper being published somewhere. And
okay, thank you so much. Yeah.
Yeah, and we're gonna have another EViNS EV club in I guess January. So until then, see you later.
Okay.
Bye-bye.
Thank you so much, everyone.
Thank you.
Thank you. Thank you very much.